1, 2, check. Everyone, my name is Orenbar. Nice to meet you all. So today I'm going to talk to you about our company, Quant Health, and what we do around clinical development, clinical trial simulation. The focus of today's talk is going to be on how we use large multimodal data sets and deep learning techniques. I'm the co-founder and CEO, as well as CTO. For that, I was the co-founder of Concert AI, one of the largest precision oncology companies in the US. I have a bachelor's in biochemistry, a master's in computer science from Boston, and the company is based in Israel, about 20 people today. Been around for about 3 years. So I'm going to continue some of the things that Professor Shai talked about earlier around the insights and data gaps, but focusing on a particular problem, which is the failed clinical trial problem in the industry today. And so a little bit about the economics of the space, just to put things in perspective, how big the problem is. So today it takes about $2.6 billion to develop a drug in around 12 years from discovery to approval. Pharma spends about $200 billion on R&D, and about half of that goes to clinical trials. Clinical trials, for those of you who don't know, are separated into 3 phases. You have phase 1, phase 2, and phase 3. Phase 1 focuses primarily on safety. Those are small cohorts, typically just to make sure that the drug is not toxic. Phase 2, typically a little bit larger, a little bit longer. The primary goal there is to test efficacy. Does the drug actually do what we hope it'll do in humans in the disease of interest that we're testing? And then phase 3 tests both of those things, safety and efficacy, at scale. They can take several years and typically several hundred participants per trial. On average, about 50% of trials fail, amounting collectively to about 90% of drugs failing the entire phase 1 through phase 3 process, which is a big problem. That amounts to about $50 billion wasted each year, so about half of the clinical R&D goes down the drain. And this is particularly important in today's advancements around data and AI and how that's impacting research, we're seeing a lot of companies in the drug discovery space, and the rate at which we're discovering new targets and new drugs is increasing exponentially, and yet our ability to push these assets then through clinical trials isn't really moving. And so that mismatch is only growing, and this problem is only going to get bigger. So why do clinical trials fail? So the majority of them fail because the biology doesn't work, or at least doesn't work the way we expected it to. So about half of those that fail because they don't demonstrate efficacy. Another quarter or so of failures are due to safety issues. And then the remainder is typically around lack of enrollment. Actually, finding patients for clinical trials is also a very big problem. And then there are other logistical and financial reasons as well for failure. But again, if you look at the first two issues, efficacy and safety, again, these are primarily biological clinical concerns. And they happen because clinical trials are extremely complex. You have to consider both the biological implications and the mechanistic understanding of the drug, and then how that works on animals, and then how that scales to humans, and then how that works over entire populations. And then on top of all that, you also have competing concerns within clinical trials. You have pharma companies that are trying to, on one hand, increase, say, the market size and the overall addressable population, but that'll oftentimes increase the noise and then reduce your signal in the data. So a lot of times, a lot of these concerns are actually competitive in nature. And so balancing all these concerns in one unified platform is key to advancing the success rate of clinical trials. So Quant Health focuses on solving this issue through in silico trials. And we focus primarily on the safety and efficacy parts. Again, as I mentioned, those are the primary issues leading to failure. But the benefits are many. And I think most, besides saving pharma companies large amounts of wasted funding and bringing better drugs to market faster, we also reduce the overall patient and system burden. So a lot of clinical trials that proceed shouldn't be going forward, and we're putting patients on drugs that they shouldn't be put on. And then also, overall, a system that is more efficient with its resources. Right? We have a limited number of patients, a limited number of hospitals across the universe that we can actually work with to run these trials. So how can we integrate technology more efficiently to run these trials using less patients, less real resources, and more digital resources instead, in order to, again, reduce the burden, increase the throughput of drug development. So to do this, Quant Health created its own data integration system. And the idea is that in order to solve this multimodal problem, we need to integrate data sets that have not been integrated before. And so this was an enormous undertaking for us, and it took us about 2 years to get this right. But fundamentally, what we've developed is a framework that combines the biological on the top, the clinical in the middle, and the epidemiological domains on the bottom. So on the top, you can see, this is, of course, a simplification of a large knowledge graph that we have, including several 100,000 entities, including therapeutics, targets, genes, cell processes, anatomies, diseases, side effects, and millions of relationships linking those entities and understanding how they impact one another. On the clinical side, we've sourced over 350 million patients, mostly from the US, where we have longitudinal data spanning over 10 years of history. And for each patient, we have over 10,000 data points. For each drug on the top, we have about 5,000 data points. And then so by linking those two data sets, essentially we can create machine learning models that, on one hand, that understand how drugs work at a very deep level, understand how humans behave clinically, sort of linking the genotype and the phenotype. And then by combining those two things together, we can learn how different drugs impact different patients in terms of real-world outcomes. By adding a third data set on the bottom of clinical trial results, we can sort of denoise a lot of the bias that exists in the upper two domains and sort of tie that back to aggregate results and clinical trial measures. So this is an AI talk, so I couldn't resist to show you a little bit about our AI architecture that we've been working on for the last several years. Essentially what you see here is the integration of those first two layers. On the top you have a knowledge graph. We use graph neural networks to create these drug embeddings. We use a separate embedding model to actually create drug-drug interactions. We can also model many other interactions as well. This is just an example. On the bottom, you see the longitudinal data set of patients. Again, we use sequence models in this case to create longitudinal models to sort of model the patient as they go through their patient journey. Combine that with an attention layer, and then all that combines with the drugs. We sort of look at given a patient and their journey, and now a new drug that we're considering to treat that patient with, what is the outcome going to be for that patient given that drug at a certain point in time in the future? And so we can predict things like overall survival, disease progression, adverse effects, and many other clinical metrics. So what does the learning process look like? In this case, you can see this is a knowledge mapping, I think, of blood cancers. So you see on the right is about, it's a bit not very crisp, about 50 different disease types, different blood cancers. You see how the different diseases sort of overlay in high dimension. And then you can also see, in this case, these two drugs, elotuzumab and lenalidomide. Those are two drugs typically given together across many of those diseases. And so the model essentially learns basically how drugs, say you have a new drug, how is that drug similar to other drugs that have treated other diseases or other patients that look like the patient or disease that I'm treating right now. So it sort of tracks all these different relationships and similarities in high-dimensional space to create these inference models. So given all the fascination and hype that's happening with ChatGPT and large language models, I wanted to show you a little bit of what we can do with this technology today. And this is something that we've just started to play around with. But the question is, how can we leverage these generative language models to help understand and explain how some of these really, really complex models are actually learning and what they're doing under the hood. And so in this case, we took a simple example. In rheumatoid arthritis, we said, let's take dexamethasone and understand what is the MOA, the mechanism of action, behind that drug for that disease. And so we took our knowledge graph, we queried all the relationships that map from dexamethasone to RA, pulled out those paths, and then fed those paths into a fine-tuned language model and asked it to explain us what is actually happening there. And the result is quite impressive. It's on point. Yeah, that's one interesting use case, I think, that we'll be seeing a lot more of in the industry. So going back to our clinical models, how do we actually make sure that these models are correct, right? So again, we're predicting how patients will respond to new drugs in a real clinical setting. That's a very difficult problem to solve. So first, we check for each model that we build, does the model work the way we expect it to? So we look at basic SHAP scores and feature importances and basically try and get an understanding of whether the drivers, the features that we expect are what actually driving the models in the way we'd expect them to. Of course, we do various machine learning assessments around F1 scores, AUC, ROC, accuracy scores. Our models, on average, are able to predict patient-level outcomes with about a 90-95% accuracy per binary endpoint. So like predicting if a patient will respond to a drug within the next 60 days. But the most interesting validation would be, of course, number 4, when we actually can compare our simulations to real-world clinical trials. We're not there yet. We're still a pretty young company, and the companies that we've helped simulate their trials are only now just beginning their clinical trials in the real world. So the next best thing that we have is backtesting, where we actually take our simulations and compare them against previously completed trials and compare the outputs. And so I'm going to show you 2 examples here. This was one, I believe this is a phase 3 trial in oncology. You can see on the left, this is the Kaplan-Meier results and the accompanying statistics of the real result. On the right, you have our simulations. For those who are not familiar, what you're basically seeing is on the bottom graph, for example, you have overall survival. So as time goes on, we're looking to see how many patients are still alive on one drug versus the other. So the higher the graph is, the better. And you want to see basically separation. So the more separation there is, where hopefully your experimental drug is above the other drug, above the standard of care, your drug is actually doing better, and then hopefully that leads to approval. So you can see on the top the progression-free survival. That was a success because the above drug actually did better. We were able to detect that signal as well, including confidence intervals, p-values, all quite similar to the actual trials. On the bottom, you can see the overall survival was not significant. We detected the same thing. Another trial, this is, I think, also a phase 3. We do phase 2, phase 3, and we're now doing phase 1 as well. In this case, you can see also success on the top, failure on the bottom. In this case, our success was actually a little bit less significant than the real trial. We can't get them all, but we get quite a few right. We're about 85% accurate on the endpoint level. So this is kind of an example of tracking one drug through the approval process as it jumps from indication to indication. You can see here we got almost all the trials correct. So is all we're doing just predicting whether a trial is going to fail or succeed? No, we're doing much more than that. So what we want to ultimately do is optimize these trials and figure out how can we do better. And if we know that a trial is going to fail, can we turn it into a success? And so in this case, this is an example where a clinical trial actually failed. This was a massive phase 3 trial. It was a pivotal phase 3 trial. It took about 8 years to complete, almost 1,000 participants, $400 million was the total cost of the clinical trial. And it was a big surprise when the trial failed. And we could have predicted this ahead of time. And we also could have predicted how the drug could have done better, how the trial could have performed better. So we actually identified that out of all the subpopulations in the trial, we indeed identified that there was either no signal or a negative signal, one where the drug actually does worse than the standard of care. But there was one case, the amyloidosis subgroup, where the signal was flipped. And so that is an important research hypothesis that we could have then tested further and see if there's actually some correlation between that subgroup and positive response. Another example where a trial actually succeeded, we then want to say whether the drug could be used differently, better in other indications. And so in this case, we identify that the drug in question could actually be used better if prednisone was added as an additional therapy. And we also identified that the combination could work well in additional indications. So what are we doing with this? How are we actually delivering these results? So we built a hypothesis testing engine that basically makes this technology and these models and data sets available to an easy-to-use platform where research teams can then run these simulations live and then select their cohort, their treatments, different comparators, different endpoints that they want to measure against, and then get the full readout, the full result of that virtual clinical trial. So instead of waiting 5 years and spending hundreds of millions of dollars on a clinical trial, can we run it in seconds and do that thousands of times across different variations to really figure out what works best? This is an example where we use this platform with one of our customers recently, a pharma company. And so this company is developing a drug for acute respiratory distress, basically COVID. And they had a clinical trial that they wanted to go forward with, and they asked us to simulate it for them, to actually test it in silico first before they go forward. And the results weren't very promising at first when we ran the simulation. And we realized that with a few fairly simple modifications to the protocol, we could make some pretty big improvements. So for example, we recognized that the drug wouldn't work just on virally induced ARDS, but it would work well on sepsis-induced ARDS, and basically all other ARDS subgroups would benefit equally, or almost equally. We also recognized that the drug would be more effective if it was given when the patient was actually undergoing intubation. So not the original hypothesis was to treat patients when they enter the ICU. Turns out you want to target the more advanced cases right when they're undergoing intubation. The customer also thought that the majority of the drug's effect would be felt long-term, given its anti-inflammatory properties. Turns out that the effects would actually be acute, and most of the effect would be felt in the first 90 days. So this means that we could reduce the overall trial length, which is quite important. A lot of trials are going to fail because they run out of funding or it's just too expensive. So all said and done, we were able to make these amendments to the protocol to increase the population size by over 4.5 times, which increases the amount of patients available for recruitment, as well as the overall market size for the number of patients who could actually benefit in the end. We reduced the overall length of the trial by almost a year. Because we can also detect the significance of each arm, we can also detect how many patients are actually needed for the trial. And so we realized that we could recruit almost a third less of the patients that they were going to recruit. And then, last but not least, we were able to increase the efficacy by almost 17%, a metric which is usually inversely correlated with those other three. So actually optimizing all of those 4 aspects together is crucial and extremely difficult. So one of the things we're doing today is we're working towards a system which can not only test our hypotheses, but also help us understand which questions we should be asking in the first place. So using our knowledge graph to help inform on drug repurposing and target validation and basic indication selection, all the way through to running high throughput screening on our clinical engine, to being able to understand relationships across a large number of phenotypes and genotypes and different drugs. And that's it. Thank you.
Works now? Okay, perfect. Hi, everyone. Thank you so much for the opportunity to speak. My name is Doron. I am a co-founder and the CTO of Wild Biotech. And I won't be talking about clinical trials. I'll be talking about translating wildlife resilience into novel therapeutics with AI. And before we dive in, I want to tell you just a little bit about Wild Biotech itself. So we were founded in 2019 in Israel by Neta, Ido, and myself. We're a seed-stage startup with 10 full-time employees, and we have a big vision. Our vision is to leverage extreme biology to develop drugs to treat a range of diseases. And I'll be telling you today about some of the things we do to try to achieve this vision. So we have four steps, 4 chapters. I'll start with the two sides, the two ends of the story, from wildlife resilience, from our basic hypothesis, all the way into the actual drugs that we develop. Then I'll talk about what's in between, how we actually leverage AI, and specifically big model AI, to do these amazing discoveries. And specifically, I'll mention how AlphaFold helps a lot. Animals are amazing. They can do all sorts of amazing things. They can hibernate, they can resist pathogens, they can eat rotten, full of toxin meat, and survive and thrive even. So how do they do that? And despite having quite similar physiology to ours in many cases, and very similar immune systems, they were evolved in very different environments, in extreme environments in which their immune system was constantly hyperactive. So we had this thought that given the tight interaction between the microbes that live inside them and their immune system, maybe the microbes evolved ways to interact with the immune system in a beneficial way that we can try to take and adapt and to transform to human therapeutics. So we went all in. We started this crazy research in collaboration with Professor Segal from the Weizmann Institute. And we basically sent expeditions all around the world to collect animal feces. And some of this amazing effort was published in Science a year and a half ago. So just to show you a couple of pictures, so those beautiful primates, golden bamboo lemur and this amazing gorilla. Or you can imagine the team actually waiting for a hyena in Tanzania to finish its breakfast until we finally have a fresh sample. So this is how a successful expedition looks like. This specifically is from Falkland Islands. You can imagine how hard it is to get to Falkland Islands. Nevertheless, we did that. And with dozens of samples, we pack them and send them back to Israel for sequencing. And we basically extract the metagenomes of those samples, and we analyze them. And we discovered fascinating things. For example, we built genomes out of those samples, microbial genomes, and we saw that more than 75% of the genomes are novel, which is like an amazing contribution to the microbial tree of life, which you can see here on the left. It is a phylogenetic tree of all the genomes we constructed, and everything in red is novel. We discovered many interesting things, like there's a phylum of Verrucomicrobia with a subclade which has completely different functional profile than other members of the phylum. When you have this broad landscape of microbiome, of wildlife, you can start seeing these interesting patterns of groups of microbes that live together. And we identified those groups of small ecosystems that tend to exist or to not exist in specific groups of animals. So you can see this, there is some complicated, interesting biology behind this behavior. And this is not surprising. I mean, we know in the sense of human health and disease, we know that many immune diseases are linked to dysbiosis of the microbiome, gut barrier dysfunction and inflammation. We know that these things tend to be systemic. We know that inflammation in the gut is related also to liver and pancreas. People are talking about the gut-lung axis and even gut-brain axis. So all these things are interconnected. But how wild biotech actually leverages this unique data to design and discover new drugs? So we actually know quite a few mechanisms of how microbes or how microbial proteins interact with the host. We have examples of activating anti-inflammatory signals. We have opposite examples of inhibiting inflammatory response. We have examples of transport. So we gained a huge interest in these proteins. But what proteins exactly? If we look at the size of potential modalities for drugs, we have this wide spectrum, right? On the left, we have small molecules, typically well below the size of one amino acid. And here the behavior is mostly dominated by chemistry. On the other hand, we have those huge antibody drugs. In between, we have peptides, so say up to 10 amino acids. And then we have this huge range of different kinds of proteins. But there is a very interesting range right in the intersection between where the behavior can no longer be explained by simple chemistry, where we have a very, very interesting and underexplored modality of mini proteins. They are complex enough to form interesting structures. And we decided to focus on this area. Now, we have very interesting examples in nature where these mini proteins are important. Think of venoms, for example. So venoms are essentially cocktails of many, many different small proteins, some of which tend to be very, very stable. Although if you think about the typical protein, proteins tend to be not so stable. And we have many examples of these mini proteins being very potent. For example, potent neurotoxins, neuropeptides. So we actually identified many similar proteins in our microbial database. On the left, you have examples of a defensin-like molecule and CRISP-type molecule. In both cases, in red, we have highlighted cysteines, which are important. They're one of the most common ways for nature to stabilize a protein with covalent bonds in the form of disulfide bridges. We have another examples where we have partial structural resemblance of phospholipase 2, also a very important venom toxin with medicinal applications. We have another interesting example when we actually we identified a neuropeptide analog, we modeled it, and we saw that it has the potential to be more potent than the homologous variant. We identified many more of these scaffolds in the database. For example, in the right, there is an example I really like because it's basically a ring. So the nature took like 2 cysteines at the edges and cyclized the mini protein. And that's actually a very common technique, a medicinal chemistry technique to stabilize mini proteins. And here we have it like ready-made from nature. So this modality is particularly interesting because it combines 2 very fundamentally important things. On one hand, it has the ability to be orally stable. So we have those toxin-like scaffolds. And you can think about the evolutionary background of these mini proteins. They were evolved to be functional in the gut. So they are stable against proteolysis. So it's a very, 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 very important feature of these mini proteins. On the other hand, they're complex enough, as opposed, for example, to small molecules. They're complex enough to be involved in some very complicated biology. You can think about bispecificity, for example. One of the biggest problems of small molecules is off-target effects. So they're basically not complex enough to be highly specific. And in this case, it's the material of signaling in the body. It is designed to be super specific. And we have this very desired bispecific functionality where, if you think about it, immune diseases, for example, people are trying to affect a single target, a single pathway. But the body doesn't work that way. So in a sense, this complicated bispecificity is very desirable. So in this modality, we find the intersection between these two very important features. And this is not just an idea. We found such bispecific mini-proteins. Here you can see a target, which I cannot disclose, but we can see low micromolar activity in a cell-based assay for two different targets, different but related targets of complementary pathways, which is a very, very good activity for non-optimized molecule. On the right hand, you see that there is no off-target effects for multiple targets that we tested. With some other molecules, we took another step, and we tested them on mice. So here you see an example of a molecule we administered orally to a TSS colitis mice model. So it's a very well-accepted model for IBD. And on the left, you basically see a comparison with other standard of care treatments. We have dexamethasone here and filgotinib. And we see that our molecule did a remarkable effect, comparable to other drugs in the market, even before optimization. So that's a very promising thing. And we see the same behavior at different levels. We see it in the cytokine level, in the cytokine related to the discussed pathway. And we also see it in the tissue level, when we see much less lesions, and overall the tissue was able to regenerate nicely after the inflammation was induced. Yet another example, this time bleomycin-induced fibrosis, lung fibrosis model. Here again, we observed very significant therapeutic effect where we see that the tissue almost entirely recovered. So we have a lot of promise with these molecules, and actually today we spent quite a lot of effort in advancing these internal pipelines for multiple areas, disease areas. But how AI helps, basically? So essentially, it's all about mining interactions, okay? We, as a platform company, we're after interaction prediction between wild mini-proteins and human targets at a very systematic level. We want to solve the problem in the most general sense possible. And traditional approaches, they took the following premise. They say, okay, so we have a protein, let's describe it in some numerical way. We can have amino count distribution, we can have different biophysical descriptors, and then we can formulate the problem as a classification problem. And these kind of approaches have very limited success. We actually did quite a few experiments ourselves. We built some of these models. We even reached a very, very nice area under the curve, a comparable and beyond state-of-the-art in terms of interaction prediction. But these models tend to generalize very poorly. And there are some work showing that these models actually tend to learn patterns in the data more than the actual biophysics of the interaction. And that's concerning. And so we need a more robust way to look at the problem. We need a more sophisticated way to look at those interactions. So what we actually want to do, we want to have some dense metric embedding space in which these biophysics are captured. And then in this metric embedding space, we want to look for relations between those entities. So basically, we want a large protein language model. Luckily, State-of-the-art advanced quite a lot in the recent couple of years, like for natural language processing in general, and specifically for the protein language. So here is an example of a work, not by us, of a model called Prosy. So they did very nice work. They trained a model, and they showed that when we take the embedding of these proteins and we use some sort of dimensionality reduction, we see a very nice separation to different structural motifs. So in comparison, for example, to simple edit distance between sequences where we see very poor separation, the embeddings on the right actually allow a much deeper insight into the actual structural nature of these proteins. Now, a non-trivial leap is to look at mini-proteins specifically, because these models were not trained on mini-proteins exclusively. And we actually showed that these models can be used for our mini-proteins, and they capture fundamental features like hydrophobicity and the isoelectric point, which is something you really want to know when you want to synthesize these molecules and make sure they are, for example, soluble. But can you actually capture interactions with these type of things? And we're showing that yes. We actually took several targets of interest. We collected mini-proteins that interact with them, and we looked where these interactors fall into the embedding space. And we see very interesting things. For example, in the top 4 examples, you see that these interactors, like, they group nicely in the embedding space, which basically means that all the observed interactors are very similarly, they're very similar in the biophysical sense. They have very similar features. But this is not always the case. You see some examples in the bottom where you have sometimes just a couple of clusters. In some cases, they're just all over the place, which may mean several different things. It may mean that the pharmacophore is very complicated. You have very different ways to capture the interaction. Or you may have different binding sites that are all relevant. Or simply, the embedding space is not capturing these type of interaction well enough. These are all problems that we're trying to solve. But this is the actually important thing. So all those interactions individually are nice, but the huge leap we're doing is to construct this big network of connections. We call it the wildscape. So basically, it's an aggregation of all the interactions we predict, and it's a map. It's a map of everything, of every potentially bioactive compound from our database to potentially therapeutically important human target. And this is the basis for our discovery process. We basically, you can imagine that if we want a specific mini-protein binder, it's a query to the network. If we want to avoid specific off-target bioactivity, it's again a network. It's again a query to the network. Same with bispecific binders. And we invest, like it's a core effort in wild biotech to make this network bigger and more reliable over time. But there is no free lunch. And turns out that such a wide systems biology approach, when you try to address like a very general problem with a good performance overall, you make compromises. And we basically try to develop novel drugs. So we need to have good performance in specific areas of interest. So we have a problem because on average, the performance is reasonable, but in specific cases of interest, we need to do something more. We need to improve the network significantly. So this is where yet another big model AI comes into aid. And this is where we find AlphaFold super useful. So AlphaFold needs no introduction. We all saw the remarkable performance in CASP 14 a couple of years ago. We all seen the Eva Former architecture doing remarkable things in terms of accuracy and speed of inference. And we actually saw that while AlphaFold originally was trained on monomers and was designed to predict the structure of monomers, people very quickly started to explore how to leverage this amazing tool for interaction prediction. So the first step was actually to formulate a binding problem through a folding process. So there is this work from an Israeli group. They said, okay, so if we can take like a single chain and fold it, basically the physics of interaction between proteins is very similar to the physics of the folding itself, essentially. So what if we just take the initial chain and we concatenate it to a long link of, say, glycine amino acids, which are very flexible, and then we have the ligand, the small protein that we want to bind. So the folding process itself will essentially mimic the binding. So we have a docking tool formulated as a folding problem. So it worked quite nicely. They actually showed that the performance is comparable to state-of-the-art flexible docking, which is surprising and very encouraging. And a couple of months after, DeepMind themselves released an updated version of AlphaFold, which is called AlphaFold Multimer, which the network was retrained to form multimers. So that's a good news for everyone trying to do protein-protein docking at scale, because this method is much faster than traditional docking without a huge compromise in accuracy. So we actually deploy these methods quite a lot in our pipelines. So one example is a situation where we have a target of interest, which has no solved crystal structure with its receptor. So we first modeled it in AlphaFold to get a hypothesis about where things are supposed to bind. And then we experimentally determined the set of bioactive binders, and we needed to prioritize them. So we used AlphaFold to try and estimate the binding energy that we expect. And these are two extreme examples. So in one case, we see a very strong binding. Some of these bindings of these contacts are hydrophobic, which are important for specific binding. And on the other case, we have a very weak binding energy. So this is a very useful tool. But where it really shines is actually in combination with the previous approach. So that's our current paradigm. We basically use the large protein language model to create a landscape, to create sort of a prior for the binding landscape of different targets with different mini proteins. And then we select their areas of interest, which can be driven statistically of relatively poorly mapped regions or high variance regions, or they can be chosen strategically, what makes sense for the business of the company. And then we use this as a navigation for structural modeling with AI. And we introduce refinement. And this is an iterative process, which makes the network much more accurate over time. So this basically finishes the entire story of how Wild approaches drug discovery. So we start with those remarkable animals. We then try to assemble their genes and genomes out of the very noisy data that we sample. Then we apply large language protein models in combination with more high precision structural modeling in a sort of active learning manner where we iteratively improve the landscape. And the resulting landscape is really the basis of all our applications. That's our, so to speak, app store, on the basis of which we build all the drug discovery applications in-house. And this is what enables those remarkable mini proteins to be discovered. And we believe down the line, they will be a key to solve many diseases that are extremely hard to solve with more conventional way of thinking. So that's it. Thank you. question.
That was beautiful and really exciting.
Do you want to join? I think I'll get it off.
Okay, I would actually start with a question to Doron. So the mini proteins, it seems that you are thinking about them as a discovery process.
Yeah.
Doesn't it make sense to think about them as from design perspective?
Yeah, it's a great question.
Okay.
You're right. So you actually touched essentially a prioritization problem that we're having. We definitely believe that down the line we will use the models that we train for design purposes, but this is a more sort of medium-term plan. At first, we're really after this bioactivity. We want to make sure that the things we find in nature are interesting enough, and the grand vision is to really insert generative design into the process based on this evolutionary approach. This is, in fact, when people ask me, like, why do you even bother? We have all those de novo tools today. You have the Baker Lab that designs drugs from scratch, and it just works. So this is not really the case. So the success rate is quite low. People don't really cover all the search space that there is to cover. So many assumptions are made. For example, we either constrain ourselves to very small constructs, or we bias the search space into known things. So that's exactly the thing we're trying to avoid. And the premise of what we're trying to do is that we can gently observe what nature has done and come up with design principles that will help us to design better drugs. But one step at a time.
Thanks. Can I also have a question to Or? I'm trying to understand the kind of what happened below the hood when you make those predictions. And I'm trying to think in a very simple case. When I look at survival curves of different drugs, let's say for the exact same indication, let's say I have cancer patients, some were treated with one drug, some were treated with another drug, and they have different survival curves, and that data get fed to the model, and I guess then I'm trying to extrapolate from there. The model probably makes connections. That drug is similar to another drug, and then I can start making predictions on efficacy. But my question or worry or what I'm not understanding is, at the end of the day, the basic data of two drugs in the clinic have different survival curves it's difficult because the very reason for giving those different drugs was the clinical presentation of the patient. And some of it is exactly some assessment of what is the survival potential of the patient. So how do you disentangle those co-founders?
Makes sense. But instead of explaining how we disentangle and reformulate the features into a predictive model, let me give you an interesting example that I don't have up here, but is very relevant to your question. So we were working with a customer. They had a drug that recently launched in diffuse large B-cell lymphoma, typically a fairly aggressive blood cancer. And what we were seeing is they came to us because they were recognizing that their drug was not being given according to the indication for which it was approved. They were seeing selection bias in the real world, where because it was a new drug for which there was not much evidence, physicians were typically giving that drug to patients that were more advanced and more severe, higher stage, higher comorbidity. Okay. And so naturally, as you would expect, the drug also showed worse outcomes than the standard of care, and much worse than the clinical trial showed. And so what we wanted to check is see whether we could debias that selection bias. And so we took all the patients who were given the other standards of care, who were of lower stage, of lower comorbidity, and basically test now that drug against all the other drugs on an equal footing. And what we realized is that the drug was about 30% more effective when you eliminate that selection bias. So that is a short way for me of me basically saying is we, by training our models on very large data sets where we have high variance across the population, and further introducing aggregate results of randomized trial data, which typically, as much as possible, that bias is removed, and using those data sets to fine-tune our models, we're able to account for those different biases and population variations.
Maybe that also relates to my earlier question about causality and what can we say without actual experimentation. And to be honest, I'm sitting on the fence. I'm I'm just thinking, I want to know the answer to that, but I'm not sure that. I think it's kind of collectively maybe something we should discuss. But I'm just trying to understand, when you say we're going to judge those two, the efficacy of those two drugs on equal footings, it means that whatever confounding factors may have existed, the model have access to those confounding factors. So if the physician was kind of looking at the eye of the patient and making an assessment based on, I don't know, the voice or how they look or something that was not written somewhere, then these factors by definition cannot be accounted for, right?
So I mean, you're stepping into a philosophical realm here.
No, not so much philosophical. Not so much philosophical. I'm trying to say this kind of a prospective study in which I say I'm going to look at the efficacy of two drugs on equal footings in the sense of in the classical sense of here is a cohort of patients I'm randomly going to choose, and it's going to be double-blinded, and one group is going to be treated with one drug, another with another drug. Okay. In absence of that, I think what we're trying to do is making such a controlled study from analyzing data, and then equal footings means we're going to compare two patients that look the same in everything that we have measured on them, but not in things we cannot say what was not, that they are the same in things that were not measured.
When I say equal footing, I literally mean the same patient, giving them two drugs, creating two arms from the same patient, which is something you can only do in silico, right? So we can, if we want to, we can completely eliminate the effect due to variance by having both arms be completely identical. So the same 100 patients in one arm are the same 100 patients in the other arm, because we virtually give them a different treatment.
Okay, we should continue offline. Thanks.
Hi. Thank you very much for these wonderful presentations. My question will go to Dr. Levin regarding the use of AlphaFold in order to predict the interaction between proteins. I found actually your idea wonderful to use the protein large language model on top of AlphaFold, because that's what we need to limit the space of solutions. The question is how we can trust AlphaFold, which is basically was designed for protein folding, to predict the folding of the protein, to do the prediction of protein complexes. Of course, you mentioned biophysical processes. And do you have any quantitative evaluations or even qualitative evaluation on some cases which showcase that we can trust AlphaFold, and on top of that, your large language model to validate the solutions?
Yeah, great point. So I'll first mention that while the original version was indeed trained on monomers exclusively to train to fold into a single structure, there is a version to actually fold multimers. So which was assessed with structures that were not part of the training set. So we have a good ground to believe that this task is reasonably is solved reasonably well with this tool. You are touching one of the challenges we have as a small platform company that tries to develop a drug at the same time, is that we need to carefully choose our experiments. So it's a trade-off of whether you take your most promising compound as deep as possible through in vivo studies and PK/PD and so on, or you do a more broad validation of the predictions. So I have no easy answer for that. We try to navigate between these two. Many of our validation processes go directly to function. So for example, we have a hypothesis about the structure. We take 2 steps, we leap 2 steps, and when we ask whether we observe the desired function in a cell-based assay. And if we don't, then maybe there is another reason to why we don't see it. But it's a guideline. I think over time we will gather more and more data to be able to validate these approaches. But so far so good. Like it's fruitful enough, I would say. And that's the bottom line.
Yeah, again, congratulations on this nice idea to use the protein large language model. I think it's time to introduce this kind of priors in order to make more accurate prediction on the interactions. Thank you so much.
Definitely, thank you.
Thanks. I have 2 questions. One is for Dr. Levin. And so, sounded like interesting. You study so many, microbiomes of so many species. Do you think you can find some antibiotics because they eat all those messy stuff and they can survive, but a human would die off? And then, do you think that would be one approach?
Oh, definitely yes. So actually, one of the first projects we did was to identify an antitoxin novel enzyme. But the sad part is for 20 good ideas, we don't do 19. And that's very unfortunate. Like, we're a very curiosity-driven company, and it is always like sad to leave those good ideas aside. Okay. Specifically for antibiotics, we believe that perhaps for us as a company is not the right bet commercially. There are many difficulties in the market of new antibiotics. But we try to leverage those ideas through partnerships. That's the philosophy.
Okay. I also have a question for Dr. Yimbar. You show several real clinical trials and then you do the simulation, right? And are those predictions have anything? You have the genomic or the transcriptional data of the patients enabling you to do the prediction, or you're just thinking the clinical data themselves is enough for you to make the simulation?
So it depends on the disease, and for diseases which are more genetically driven, we typically, at the moment, we're less focusing on those because we don't have deep sequencing data, but we do have sequencing results. So we have biomarkers, sometimes even SNPs, all of that out of the EMR. So the data that's in there gets quite deep, and it's improving each day. So I think there's a point where we'll have all of what we need, I think, for the vast majority of cases.
All right, thanks.
Hi, question for Or. So you spoke a lot about your platform, which is based on data-driven predictions, and I'm sure that there is also a lot of domain knowledge coming from experts for its clinical trial, coming from the actual, institutes running this trial. And I was wondering how you integrate these two sources of data together.
So good question. So we have a baseline framework which integrates all of the data sets that Quan Health has developed as its kind of proprietary integrated data set. This includes clinical trial data, patient-level data, and biomedical data about drugs and pharmacology data. That framework is quite flexible, and we can also take additional data that was developed by pharma companies. So say preclinical or phase 1 data, we can actually take that and layer that into our framework and add additional evidence to the simulation.
Is there a principled way to do this integration, or do you kind of tweak the model on a case-by-case basis?
So it depends. So like for preclinical data, we would do some form of like allometric scaling or PK/PD model overlay. And so that's quite different than how we would say integrate phase 1 data into a phase 2 trial, which already has human level data. So then we would link the data and essentially provide, basically stratify our existing patients by biomarkers or features and outcomes that were discovered in the previous phase.
Got it. Thank you.
Thank you for talk. I'm quite new in this field, and I want to ask the question. I suppose that collecting the database is the most important part and is the difficult. And my question is, could you give the advice or key rules to how efficiently collect the database from animals?
Wow. I'm afraid if there is an efficient way, we haven't figured it out. It was a very, very tough endeavor. Basically, I think maybe one advice I can give you is like try to see the bigger picture. Like for example, I can give like 2 examples that if we were thinking about it beforehand, it will make our lives a little bit easier. So one thing is to try to choose like a focus group. For example, when we just started the collection process, our hypothesis was that there is something interesting in this enormous wildlife microbial landscape, and we don't know what it is, so we want to collect as wide as possible. But as time went by, we as a company, we developed this focus on developing therapeutics, which was not there in the first place. And then it was much clearer that we want to focus on mammals, for example, because of the resemblance of the immune system. So perhaps defining the what are you trying to achieve with the data as early as possible is crucial. I guess it goes without saying, but in a scientific setting, you're not necessarily driven by these things, and you're more driven by exploration. So I think it's a comment worth mentioning. And another thing is just to try to talk with as many people as possible that try to do this, and try to break their paradigms. So we had like 1 expeditions where we were after whales, and the people there told us that it's basically impossible. So they're researchers like trying every season again and again to capture these whales and to extract species, and we just didn't agree that this is the case, and we pursued them, and we get our whale samples in 1 expedition. So just try to break paradigms when people tell you that it's difficult. I hope it's useful.
But it was easy.
What?
Like you collected the data, like from the animals, it was easy. Like you broke the people's paradigm.
No, it's never easy, I think, but you need to be persistent.
Okay.
So I'd actually like to ask a question I'd be interested in the opinion of both of you. I think you're both using a lot of data. You're both using, building on AI methods and techniques. Could you speak a little bit about what you think is the next for the problems you're each working on? What type of data would be essential for driving? What type of data is missing that would be essential for the problems you're working on? And then conversely, once you have that data, what are the AI techniques that where you see the challenges and the next developments that are needed? And then maybe this is also for both of you. Maybe this is more for Or, but it seems Doron is developing small molecules that they think could be effective as drugs for some diseases, and you're developing the ability to basically take those and predict the outcome of a clinical trial. So what is the gap, or can you already take his molecules and make predictions? And if not, then what is the gap, and what would you need in order to do that?
Okay, about desired data sets. So I'll maybe comment that one of our most fundamental problems is that okay, so we are very interested in interaction databases, and these are so poorly standardized that it is extremely hard to combine, to have a data set which is both large and high quality. I guess it's like a basic problem in any application in this room. And the problem becomes even more severe when we have a focus group. So we are extremely interested in this mini proteins range. And data for this is even more sparse. So I wish we had a very reliable way to generate high throughput interaction data, which we currently don't. We produce a lot of experimental high throughput data, and we invest a lot to increase its reliability, but it by no means a solved issue. I think that the next natural thing is a data that basically more related to how these no, you know what? That's all.
And in terms of methods and
so definitely pre-trained large language models is key. I didn't mention verbally, but the language model I showed, it took 51 days to train. So it's basically impractical for a startup like us to retrain such a model from scratch. So we do benefit from the improvement and scale of hardware, but it is almost negligible in the time life of a company like ours. So we benefit a lot from existing models and the ability to retrain them sort of slightly. So I would say that the field of transfer learning is super important for our applications.
So for the data question, I could talk for days about how data is dirty, disconnected, and siloed. But I think the industry is working on that and making progress. I think when we look though at the fundamentals, I think what we're really missing is behavioral data. And so all the data that's being collected today, the majority of it is fairly pristine, empirical data about basically about the biology and about the epidemiology. And then, but we're missing what patients who are born with the same genetic makeup, what happens to one person who lives in the US and another who lives in Beijing, or where perhaps there's more pollution or different eating habits, different healthcare system. And we often miss the nature versus nurture question, I think, when we're doing this highly kind of pristine research. And so the more that we're able to include that sort of data, such as patient-reported outcomes, telemetry data, a lot of microbiome information as well, I think that will explain a lot of the variation that's currently not explainable. Regarding potential collaborations, I think we should talk. We do actually work in the sort of bridging the gap between preclinical and clinical to sort of be able to progress drugs further through the pipeline. We have various use cases that we're exploring with large pharma companies, as well as with AI-driven drug discovery and drug development companies. I think it's, as I was saying in my presentation, I think it's a must for companies that are developing drugs with high throughput capabilities to project that high throughput capability further through the clinical phases as well. We're also just going to hit the same bottlenecks we've been hitting over and over again. With regards to new AI technologies, yeah, I think having standard pre-trained models is what we're seeing. I think is the big game changer. Whether it's a language model or a protein model or even vision models is less important. But having standardized data sets on which we can both train and then fine-tune and then predict across various institutions and different data sets is key, together with then the training and release of these pre-trained models.
I've got a question out of curiosity for Or. In trying to predict a clinical trial outcome, obviously it's a hypothesis generation rather than hypothesis testing, but have you had an opportunity to compare your approach versus, say, Mendelian randomization? That's a human genetic approach. It's got some very nice principles then as to, with the kind of, with an underbedding of randomization and there's lots of nice examples of how Mendelian randomization can pre-predict RCT outcomes. Obviously, one would never rely on that entirely, so you still need to run your phase 3 trial. But out of curiosity, have you had, or what might be the pros and cons of that to meet a naive person of your own approach for RCT prediction versus an approach such as Mendelian randomization?
So I didn't quite catch the essence of the question. Could you summarize it for me?
Oh yeah, so an approach that's of huge interest to clinicians, and so is trying to use observational data, large biobanks, then using a genetic approach called Mendelian randomization, whereby you've got a genetic variant that's associated with, let's say, LDL cholesterol, and then looking at its effect then on, let's say, cardiovascular disease outcomes. So you get genetic variants that are associated with LDL, and then try and see essentially people who are genetically loaded to have lower LDL levels, do they have lower outcomes of stroke or major cardiovascular disease episodes. So essentially using an instrument variable approach, but the nice a property then to the genetic approach is that by the process of meiosis, there's this random allocation of alleles, and so it's kind of got some nice randomization properties to it. And there's a lot of interest in the biomedical field around this to try and prioritize targets then for drug trials. It's kind of analogous to what you're doing then, but quite a different approach. And I was just wondering where the two might sit versus each other, but very happy to take it offline if it's a bit more complicated than that.
So I think the two approaches are quite complementary. And I mean, we follow the same paradigm, right? We look for, on the hypothesis testing side, we look for various patient characteristics, for example, that correlate with the disease, and how can we modulate that, and then how can we target that, and then put that into our trial. And so we also randomize, right? When we say design a trial for 200 people for type 2 diabetes, we have tens of millions of patients to select from, and we select them randomly. And we can also select them not randomly, right? And we can introduce bias in a way that perhaps would more accurately model a trial in the real world. Say if we're modeling a trial in Europe or in the US on Caucasians, then we could potentially formulate the population according to that, or we could create a more equally kind of diverse population, both genetically and phenotypically. And so I think the best of both worlds will be when we have everyone fully sequenced with all the clinical data and all the behavioral data that I mentioned, and then all of that is now available both for modeling to create higher precision, higher recall models, but also giving us greater resolution on stratification and cohort selection.
Okay, let's thank both speakers again, and we have lunch just outside right now. Thank you.